Hello, everyone. Welcome back to my single-cell proteomics video tutorial. In the first video, we know the simplified demonstration data and created a SCP features object. So you can see we have 4 assays in this dataset. They are PSM dataset. So today I'm going to show you how to process the PSM dataset and the aggregate PSM dataset into peptide data. So we don't need the MQSC data and also the sample annotation anymore. Let's remove both datasets. Now we only have the SCP object. So all the single cell sequencing data, DNA, mRNA, or peptide, contains 0 value in the matrix data. The 0 values could be biological zeros or technical zeros. The biological zero means the protein is not expressed in this cell. Then the peptide sequence of this protein is 0. The technical zero means the protein actually is expressed in the cell, but the peptide sequence of this protein was not detected during the data acquisition. So the first step for the PSM data processing is to replace all the zeros by NA. So for the demonstration data, we have 4 assays. If you want to use the full dataset, it has 177 assays. So you need to replace here 1 to 177. So let's replace all the zeros with NA for this demonstration dataset. Then next we need to filter out no confidence PSM data. So I already showed you the data contains a reverse peptide column and also the potential contaminant column. They are indicated by a plus in the column. So we can remove both labeled reverse peptide and the potential contaminant peptides. And also we have the spectral purity. We can remove the cells that don't show the spectral purity and also only keep the spectral purity bigger than 80%. So let's do the first filtration. So you can see the reverse peptide and also potential contaminant are found in all 4 assays, and also cells has the purity below 80% in all 4 assays. So next we can filter the assay based on the detected features. So according to the online tutorial, the detected features below 150 could be a blank runs or a failed runs. So let's have a look at the detected features for this demonstration dataset. So you can see we have 4 assays, and the 11 and 16 stands for TMT 11 or 16 intensity reports. So this dataset, they actually first use the TMT 11 method to connect the data, but in the middle, their machine was updated to TMT 16, so they decided to connect the rest of the data using the TMT 16 machine. So you can see in 1, 3, and 4, the detected features are more than 150, but the second assay, the detected features is only 60. So we can keep the detected assays bigger than 150. Now we can filter out the features below 150. So if we have a look at the SCP object again, now you can see we only have 3 assays. So we filter out the second assay because it only has 60 detected features. We are going to do more filtration. So the third filter features is based on the sample to carrier ratio matrix. So let's have a look at the sample type again. You can see we have the blank carrier, samples, macrophages and monocyte, reference, and unused channel. So the sample channel will be macrophages and monocyte, and we have the carrier channel. As I explained in the first video, the carrier channel contains 200 cells. To boost the peptide identification, we are talking about the single-cell proteomics. In the ideal situation, the sample channel should just contain 1 cell. So the best sample carrier ratio should be 1 divided by 200. So let's calculate the sample to carrier ratio. The column variables are the sample type, and the carrier pattern is the carrier, and we know the sample pattern are macrophages and monocyte. So the function will be the mean, and also we give a raw data name as the mean SCR. So let's calculate the SCR for 3 assays. Then we can create a figure to see the distribution of SCR. And we use the ggplot package, and we can draw a line for the value of 0.005 and another line at 0.1. So let's create the figure. We can zoom in. You can see the best distribution is around 0.01, and the ideal value is here 0.05. According to the explanation of online tutorial, the SCR data was shifted to the left and indicate something wrong during the data acquisition. So the online tutorial used the 0.1 to filter the SCR features. So we can filter the data, use 0.1 for the mean SCR value. So I forgot to explain the full data for you. So after the filtration for detected features for the full data set, we only have 173 assays in the full data set. So in both steps here and here, you need to use 1 to 173 if you want to analyze the full data set. So we are going to do one more step for the data filtration. This step of filtration is based on the filter features to control for false discovery rate. So the online tutorial suggests to convert the PEP value to Q values, and after the calculation, the Q values is directly related to the false discovery rate. So let's calculate the Q values for each assay, and we only keep the cells that have Q values below 1%. So let's do the last step of filtration. Because we are going to aggregate the peptide data and later join all the peptide data into one peptide assay. So to make it comparable between different data sets, we need to calculate the relative report error intensity for each data set. This step can correct the variations between different assays. You remember, for the sample type we have a reference channel, and each reference channel contains 5 cells that are designed to normalize the data between different assays. So we can divide the sample channels by the reference channel to correct the variations between different runs. So we processed the PSM data set now. Now we can aggregate the PSM data to peptide data. Before we do the aggregation, let's have a look at the SCP object. You can see at the moment the SCP object only contains 3 assays, and they are the PSM dataset. Now we can aggregate the PSM data to peptide data. For the peptide aggregation, we use the function aggregate feature over assays. So we have 3 assays in the SCP data, and the feature columns will be the modified sequence column in each assay. Now we can give a name for each aggregated peptide data as peptides plus original raw data names. So let's aggregate the PSM data to peptide data. We only have 3 assays. You can see it is very quick. We aggregated 3 assays. For the full data set, we need to aggregate 173 data set. It takes a while to run the aggregation. So now we aggregated the PSM data to peptide data. We can have a look at the SCP data again. You can see now we have 6 assays. 1 to 3 are the PSM dataset, and we created 3 new assays, 4 to 6. They are peptide data, so we can keep the original PSM data in the SCP object. So now we can use the plot function to plot the SCP again. Okay, we need to adjust the size of the plot windows. Let's plot the SCP again, and we can zoom in. You can see now we have 3 PSM datasets on the top, and they have been aggregated to 3 peptide datasets. So I'm going to stop from here for today's demonstration. You can practice to see if you can process the PSM data set and aggregate them into peptide dataset. In my next video, I'm going to show you how to process the peptide data and aggregate the peptide data into protein data. Once again, please subscribe my channel if you haven't to do so. I hope to see you in my next video tutorial.